Dear children, in this module, we are going to learn about photorespiration. We just learned that photorespiration is lacking in seafood plants. So, what is photorespiration? It is nothing but respiration in the presence of light. So, it is taking place during photosynthesis. So far we know that it is a wasteful process. It does not produce sugars, and we know during respiration what is released out. Oxygen is utilized and carbon dioxide is released out. So let us learn in detail about photorespiration. So in Calvin cycle, we know the first acceptor or primary acceptor of carbon dioxide is RUBP. So carboxylation of RUBP takes place in the presence of enzyme rubisco. Ribulose 1,5-bisphosphate carboxylase oxygenase is a full form of rubisco. So RUBP plus carbon dioxide in the presence of rubisco, it gets converted to form two molecules of 3-phosphoglycergic acid. Now the active site of rubisco is able to bind to both carbon dioxide and oxygen. Hence the name rubisco. The last co stands for carboxylase and oxygenase. Rubisco has greater affinity for carbon dioxide than for oxygen. But what binds to the active site determines about the relative concentration of carbon dioxide and oxygen. And that determines which one will bind to the enzyme. If there's greater concentration of carbon dioxide, then surely carbon dioxide will bind to the active site of rubisco. And if there's greater concentration of oxygen, then surely oxygen will bind to the active site of rubisco. Now, if you see, in C3 plants, some oxygen binds to rubisco. Hence, carbon dioxide fixation is decreased. There is equal chance that the oxygen can bind to the rubisco in C3 plants, which has the C3 cycle or the Calvin cycle. Here, RUBP binds with oxygen and it forms one molecule of phosphoglycerate and one molecule of phosphoglycolate. We can see one molecule of phosphoglycerate and one molecule of phosphoglycolate is produced. This is phosphoglycolate and phosphoglycerate is formed. And what happens? We know RUBP has 5 carbon atoms, and this RUBP combines with one molecule of oxygen and it breaks down to form phosphoglycolate, which is having 2 carbon atom, and 3-phosphoglycergic acid, which is or glycerate, which is having 3 carbon atom. And this pathway is called as photorespiration. In this pathway, we can see there is no synthesis of sugars, ATP, NADPH. Hence, photorespiration is considered as a wasteful process. Rather, it causes the release of carbon dioxide by utilizing ATP. So, in the presence of ATP, what is getting synthesized? We can see oxygen is taken in and carbon dioxide is released out. So, this process is called as photorespiration. But what is the peculiarity of C4 plants? The C4 plants, photorespiration does not occur because they can increase the carbon dioxide concentration at the enzyme site. We know the enzyme is present in which part of the plant or which part of the leaf. It is present in the bundle sheath cells. So they can increase the concentration of the carbon dioxide by the C4 pathway, and thus the concentration of carbon dioxide is increased in the bundle sheath cell where rubisco is present. This takes place when C4 acid from the mesophyll is broken down in the bundle cells to release carbon dioxide. This minimizes the tendency of rubisco to bind to oxygen. So oxygenase activity of rubisco is minimized. Due to lack of photorespiration, the productivity is increased. We learned that they lack photorespiration and C4 plants also have greater productivity of biomass. So it is just because photosynthesis is taking place instead of photorespiration, photosynthesis is taking place. Only photosynthesis takes place, there will be greater productivity. So C4 plants have greater productivity and yields are better in C4 plants. In addition, these plants can tolerate higher temperature. In addition, they can survive in higher light intensities or they can thrive over higher temperatures. So this is about photorespiration and why C4 plants are considered to be more advantageous than C3 plants. Thank you.